There we go. All right, Jana, floor is yours. Make sure you unmute.
I'm sorry.
There you go.
Yeah, thank you very much for the introduction and for inviting me. I also feel like I'm among my people, as Catherine said. And Spiridon actually asked me to present a subject which has been a long-term topic of mine, and it's organic memory or organismic memory. And hopefully no one knows me here, so you won't see that I repeat myself a lot in these slides, except for Kalevi, who was an opponent of my dissertation thesis many years ago. And so I will start. Yeah, what I think what I think by memory. So I will minimize this. So if you have memory metaphors, which was the sort of introduction of my dissertation thesis from 2012, you usually find metaphors in history of philosophy, physiology, biology, psychology, quite associated mostly with the human brain, right? And there is always this deep association with concepts like storage, metrics, place. There is, of course, throughout the history, there is this long-term idea that even psychological states, for example, are physically localized in our brains. And it goes even to Plato with his bird cage, where birds are memories, or Augustine's treasuries or caves of memory. There is also the metaphor of a book, or as a memory metaphor of a stone, up to this Gall's phrenology, which you definitely know, where there was this idea that basically all emotions, all kinds of states of our mind are directly represented in our brains. And on the other hand, you may find memory metaphors which are more temporal, where memory is a temporal entity without any connection to a place or matter. Of course, you think of Bergson in this case, but these views, I have to say, are relatively rare in history and also like in biology. So first, I should say what I think by the term organic memory. In general, memory represents the ability or process by which a living being is able to store and later recall its own experience. And the notion of organic memory is not new in biology. It has been defined in various ways. And I'm building with this term on previous work by Walter Elsasser or Laura Otis or Marcello Barbieri. And I use this term to describe a form of memory that is not necessarily brain dependent, again, and neuronal. Rather, it is fundamentally associated with living organisms, from bacteria to humans. And I will first make some historical introduction of this term, which I find quite interesting, as these ideas are returning from time to time, because this idea can be traced back to the 19th century biology, psychology, criminology, and so on. And and I would like just to mention these works of mine. For example, you might be interested in this book on organismal agency, where also Spiridon contributed. And there is this attempt in this book to synthesize also biosemiotics, processual philosophy, or processual biology, and the ideas of extended evolutionary synthesis. So if you are interested, I can send it to you. But let's get back to the historical outline of organic memory idea. Of course, maybe you know that this idea appeared within the framework of the Lamarckian theory of heredity. I will just introduce some context here. As you may know, in 1859, Origin of Species by Darwin was published. And Darwin's theory also has Lamarckian features. There is this theory of pangenesis. Of course, Lamarck and Darwin, they weren't like opponents at their times, right? Or at the time of Darwin, as we consider them now. But between 1880 and 1920 to 30, there was this so-called eclipse of Darwinism, which means that Darwinism was not yet in favor scientifically. There were other ideas like neo-Lamarckian ideas, orthogenesis, mutationism, and so on, which were more prominent. And during this period, neo-Lamarckism comes into play for a while, right? And it's exactly within the concept of organic memory, which can be time-wise framed between these years. And it basically means that memory as such is attributed to all living matter. That means that all traits inherited and acquired are stored in the organism and transmitted to future generations. Through learning and constant repetition, the trait can become a true habit, which only leads to instinctive behavior and automatic action of the organism. So, of course, this idea was, as I said, rooted in Lamarckism, because this analogy naturally helped to explain heredity of acquired characters. However, it's fair to Lamarck to mention that although we know Lamarck nowadays for this theory of inherited and acquired characters, it's not like his original idea. He didn't emphasize it that much. This mode of inheritance was basically and simply a common way of thinking about heredity at this time. Yeah, so it was emphasized as his idea quite later. And who first attributed explicitly the faculty of memory to all organic matter was Ewald Hering, a physiologist. And he believed that all attributes of an organism, hereditary as well as acquired, are stored step by step in the organism and further distributed as memory traces available for future generations. He actually says that memory is rather a faculty of an unconscious life than a conscious one. Yeah, and also the memory of our brains, as in case of humans, for example, it represents only one concrete example of such a general faculty of organic matter to remember its experience. And also, for Ernst Haeckel, this idea was quite important as he tried to merge Lamarckism and Darwinism. He also drew an analogy between heredity and memory. And he had this idea of organism as a unique pattern of wave-like motion of plastidules. Yeah, plastidules were the units of living matter, the ultimate units of living matter, and their vibrations changed depending on the environment and the history of the organisms. Therefore, every organism eventually was a unique pattern of wave-like motion of plastidules. You may came across August Forel in terms of organic memory, who was a Swiss clinical psychologist, and when he was wondering how an embryo is able to remember into which organism it should develop, he also explained it through heredity and memory as being essentially the same. And interestingly, he claimed that both memory and heredity are based on some sort of hidden script, which is passed on and will be passed on to future generation and expressed later during ontogeny. You may know Samuel Butler also as a novelist, but he was a big fan of Darwin, but later he turned to Lamarckian philosophy and also made this analogy between memory and heredity, and they served as foundation of his panpsychist interpretation of Lamarckism. According to him, organisms are capable of remembering their experiences and incorporate them phenotypically, but unconsciously. He actually published 2 books on that, Life and Habit, and later Unconscious Memory. And he describes phylogeny as getting used to new activities, which via frequent repetitions become automatic and thus unconscious. So for him, every intelligent conscious activity eventually becomes instinctive and inherited. Yeah. So instinct is actually for him a hereditary habit. Yeah, which is quite different conception from today's ethology, for example. And he also developed the idea of memory based on vibration patterns of ultimate living particles. Also, there is and I will make it quite quickly now. That is also Théodule Ribot, French philosopher and psychologist, who believed that conscious memory is only one aspect of the more fundamental phenomenon as well. And his organic memory was rather Cartesian-like. Yeah, it was based on physics. For him, heredity is only one of the versions of the law of energy conservation, and we should describe memory and heredity in terms of work, force, energy, and particle motion. But his ideas were interestingly also like that memory is also very associative. So not only modification of cells, but also interactions between them create these memory traces. You may heard of Richard Zeman, which unified memory and heredity under one concept called mneme. It represented the basic capacity, again, an organic capacity of organic memory to maintain an after effect of stimulation as a stable modification of irritable organic tissue, and thus creating engrams, sort of memory traces. And also, for example, the son of Charles Darwin, a botanist, he also believed that germ cells in the body of the plant are telegraphically communicating within the within the whole plant body. And here you may see a lot of more examples of thinkers who either implicitly or explicitly dealt with this idea. Yeah. So you may heard about some of them. And interestingly enough, this organic memory idea can be also traced outside of biology. For example, in case of Benedict Morel, he in psychiatry, he explained the increasing incidence of alcoholism, syphilis, epilepsy, criminality as the accumulated hereditary burden, yeah, within this theory of degradation, as sort of sins of our ancestors, of our fathers. And interestingly, it was even before Hering and Haeckel introduced this idea. In criminology, Cesare Lombroso, with his book Criminal Man, he came with this idea of criminals from birth, which he perceived as lower stages of human development in sense of biogenetic law, in Haeckelian biogenetic law. They were sort of awakened atavism for him. And you can see other thinkers working with this concept also. This idea of organic memory was reflected also in writings of Emile Zola, Baroja, Unamuno, also in psychoanalytics and so on. This is very nicely depicted in the book by Lara Otis, Organic Memory. And she actually claims that organic memory was sort of thought paradigm between 1870s and 90s, 18th, and it was link between nature and culture, yeah, because when everything can be remembered, even cultural influences, it doesn't, you don't need to necessarily differentiate what is given by nature and what is given by culture. Also, an individual organism, individual being reflects the cultural development. They are analogical, and that is valid also for individual and collective consciousness. And so here are some examples of the book where you can read about this organic memory idea. And the organic memory was basically an attempt to explain evolution as teleological continuum, where each organism was a link between the past, present, and the future, participating in an immortal chain of being, where instinct, habit, and memory were identical manifestation of the fundamental capacity of living matter to store experiences. So eventually, this idea kind of lost its battle against Weissmannism around after the end of the First World War, and after the discovery of Mendelian laws, the memory theorists failed to come up with any plausible alternative explanation of how memory traits or acquired characters are transmitted to future generation. So this idea was basically abandoned. And as you may know, the modern synthesis biology did not care for sort of soft heredity or for these phenomena for several reasons. And I will now get to our view, which is, for example, expressed in this book we wrote together with Anton Markos. I mentioned this historical introduction because not only because I like it, but to some extent it's very similar to our view as we consider memory being an inherent property of the living. Yeah, because, and this also concerns individual cell, as was previously mentioned, because experience matters to intracellular processes. I would quote our book: cells do not arise by copying, and they never emerge de novo according to a blueprint. They are born and they keep the tradition of how to understand, among others, the genetic script in a given lineage. They decide, based on their experience and memory, what should be processed in what context. So how is this tradition of understanding actually maintained, except for DNA blueprint? That I believe it's through several levels of memories, which I would like to introduce here. It should be also noted that the original Neo-Lamarckians had been accused of merely positing an analogy between memory and heredity, implying that their doctrine of organic memory actually brings nothing new. Yeah, you can have like a similar objection to my presentation, but I think that in order to account for experience in organism, we should differentiate between mere heredity and memory. And thanks to the current state of knowledge of epigenetics and evo-devo, we have reached a better understanding, I believe, of the processes which constitute cellular and supracellular memory. Yeah. So if heredity is to include the inheritance of experience, which is, as I said, something not accounted for in the biology of modern synthesis, then it's better to distinguish between these two terms. Yeah? And the organic memory concept, where heredity is viewed as memory, emphasizes that organisms are historical and temporal beings, that we should account for their experience as well. Yeah? And they can learn, but they also forget, of course. So, and in the chapter of the book, in the chapter of mine in this book on organismal agency, I start with the lowest level. And although bacteria do not have complex nervous system like animals, they do have ability to store and pass on information between individuals and between generation through several processes. For example, through the process of horizontal gene transfer. I don't want to be like textbooks here. It's a common mechanism of genetic exchange among bacteria. It can be done through several ways, and it's quite widespread. Yeah, up to 80% of bacterial genes have been involved in these exchanges at some point in their evolutionary history. There is also this already mentioned CRISPR-Cas system. I don't have to explain it now in detail, but basically it allows bacteria to protect themselves from foreign genetic material, such as viruses and plasmids, by capturing and storing fragments of foreign DNA or RNA into its own genome. And these fragments are then used to recognize and destroy any future invasion of the same or similar foreign genetic materials, right? So it's basically a learning. And in this sense, CRISPR-Cas system allows bacteria to remember past infections and use this memory to defend themselves against future infections. And most importantly, bacteria can pass information to their offspring through epigenetic inheritance, which can be also taught as a form of cellular memory. Bacteria can regulate genetic expression and adapt to changes in their environment by chemically modifying their DNA. This modification can be passed to the other cells during division, allowing them to inherit this memory of previous environmental conditions and respond accordingly. Yeah, the fact that these modifications often emerge thanks to the environmental signals from the environment is important. And of course, we can see more complex epigenetic memory in case of eukaryotes. I will just briefly explain this, not to delve into details. But in eukaryotes, you have essentially three epigenetic processes, like non-coding RNase molecules, long ones, short ones. They can do a lot of things. They can regulate the sequences in the genome. Also, the DNA itself, which you can see here, can be methylated. Different letters on the DNA can be marked with sort of, let's say, diacritics of the basic text. Can be added, and it usually means do not transcribe these genes when there is this modification, but not always. And the DNA itself in the cell, in eukaryotic cell, is wrapped on the protein structures called histones or nucleosomes. And these proteins, these are proteins, not amino acids, they can be also modified. And there are like hundreds types of modification which can be found as in terms of different modification. In terms of amount, it's like millions of such modification in our bodies, in every cell. So, but the main message to take here is that all these modifications actually enable either to activate or deactivate some genes. Yeah, so you can have basically the same information, but it depends on this modification which genes are going to be activated and which will be switched off. So that's the main function. And so, and either you open the DNA from the nucleosomes or you tighten it even more. Yeah, but that's a very simplifying way to say this. It's very complex network. So, as I said, in case of bacteria, epigenetic modifications are often induced by environmental conditions. Yeah, when you have enough light, the butterfly with the same genome can change its appearance. Also, nutrition is a very decisive factor from the environment in order to influence your epigenetics. This is a mother and daughter, and the mother was fed by nutritious food, or also social interaction of Schistocerca gregaria can change the solitary form to gregarious form. Yeah, so there are more environmental factors which can influence our epigenetics in this sense. So environment is important. What is important in comparison with mutation is that these epigenetic modifications are reversible. So it can be written and rewritten again within minutes, hours, or sometimes they do not have to be erased, this modification on the DNA, for example, like methylation. They do not have to necessarily be erased during the whole lifespan of an organism. And this creates sort of metastability of the epigenome. And it actually explains why development of an individual organism is both plastic and canalized, because you can quite quickly, depending on the environmental influences and many other conditions, you can influence the genetic transcription, and you can form different phenotypes. Yeah, but on the other hand, it's canalized, so you usually end up in the expected valley of the epigenetic landscape. Of course, epigenetic modification enable cellular differentiation, so you have one pluripotent cell, and thanks to these various epigenetic patterns, you can create several several cell types depending on the same genome, and they are often inherited through several generations sometimes. Yeah, I don't think epigenetic inheritance is a problematic concept anymore. So as my point here is that the experience of an organism with the environment can be remembered or forgotten. These epigenetic modification are experience dependent, is sort of memory for future generation, allowing organism to respond quickly to environmental changes. I see some comments. I don't know. Yeah, good. I thought that it was on me. And yeah, so that's what I already said. And this form of adaptation, like epigenetic adaptation, is not random but goal-directed, shaped by the functional role of memory. Yeah. So over the course of evolution, organisms have been finely tuned to regulate their genomes in ways that anticipate the environments they are likely to be born or hatched into, environments in which they will likely encounter recurring signals. So it's sort of these modifications are sort of representations of some material manifestation of the goal-directed tendencies inherent to the organisms. In my dissertation thesis, I also developed this idea of developmental memory, which concerns ontogeny, which will be last example of memory which I will use. And we, of course, may use many more, but I focused in this presentation on these two. Because the dynamics of the developing embryo is usually explained through developmental regulatory networks. That is complex interaction between genes and their products, which are transcription factors or regulatory RNAs. And these control the expression of target genes and subsequently also formation of cell types, tissues, and organs. And such control dynamics can also change in response to various signals from the environment, such as hormones, growth factors, nutrients. And these switches can also be mediated by epigenetic modification. And interestingly, these developmental regulatory networks are very conserved across different species, which points to the homological origin of these regulatory genes. And similarities in interactions within the regulatory networks that control development have been also found in wide variety of organisms, ranging from flies to humans. I will take the example of Hox genes here. which are conservative genes shared as far back as the last common ancestor of Bilateria. They are usually called master control genes because they determine the anterior-posterior axis, and when it is formed, they further regulate the development of other organ structures, which you can see here in colors. They have role in cell differentiation, vision, death, and so on. And what is interesting, they are very conservative. They do not mutate. Their evolution follows the path of the duplication and divergence of function, meaning the same script can have different functions in the development. And so the important message here is that taxonomically distant organisms share the same developmental components, like Hox genes, for example, but not only. But difference is made in how they are used in different locations. The same genes can be used in different tissues and organs and lead to different results. How they are used differently in time, that's so-called heterochrony, for example, or for example, in terms of quantity. Yeah, even the quantity of a genetic product of a transcription factor morphogen can lead to changes. So these differences, not the script itself, leads to distinct morphologies. And I have here very simple network, regulatory network of an eye development. You see the network of where PAX6 or A, which are homologs, play a crucial role, and these are other two homologs in Drosophila and Xenopus. But you see that the wiring of the network is quite different. Yeah, that's like very simple way how to show how these networks usually change. Yeah, the actors usually may stay the same to some extent, but their interactive network changes, and it can lead to very different eyes, as you can see. But usually the networks look like this, but it doesn't tell you much if you look at this. It is a famous book by Eric Davidson, and the book is full of these developmental regulatory networks. I will just mention that these developmental regulatory networks are also dependent, as I said, on the environment, as development is not controlled only by genes, but also by the environment. And you can see two species of finches, Darwinian finches, cactus finch and ground finch, and their beaks are quite different. Yeah, so although the underlying genes and regulatory networks controlling the beak development are similar across different bird species, the differences in the diet and other environmental factors can lead to difference in beak shape and function. In this case, it is a heterometry of bone morphogenetic factor 4, which is a morphogene. And when you use a lot of it during development, you have a ground finch beak. And when there is less of this morphogen, you have cactus finch beak. So, and this morphogen BMP4, it's again very nice example of how these morphogens can be used in a different interpretative networks. They can be used for development of fingers or bones and so on. Yeah, so the same morphogen can be used as a signal for different developmental morphogenetic structure. So my conclusion on this will be that organic memory in development, as I was trying to present it here, is not a merely storage device in the form of DNA molecule. Rather, it is an interactive network, the lineage-specific usage or interpretation of the same toolkit. Yeah, you can see where biosemiotics can come into this. And of course, this specific interpretation, lineage-specific interpretation, is based on the experience with the environment, but also, of course, several other factors such as maternal input, genetic information, biophysical forces, and so on. But I argue that such networks and changes they undergo are in principle historical, habitual, and experience dependent. And I also, in my conclusion, connect this epigenetic processes and regulatory networks with plasticity, because I believe that these experience-dependent changes in terms of epigenetics and evo are a means of keeping plasticity of the organisms, meaning that all organisms are to some extent plastic, and plasticity can be observed at many levels of organization. Ranging from behavior, physiology, hormone production, and so on. And I believe, like taking inspiration from Newman, but also from others, that organismal forms are inherently plastic, and being potentially exposed to constant change, organisms stabilize their evolutionary existence through more and more fixed relationships between informational molecules, such as DNA and phenotypes, and memory is a crucial aspect of this stabilization. So this was my attempt to bring a processual interpretation of phenotypic plasticity and epigenetics and these developmental networks. So this is, of course, just only part of the whole story. I believe we will hear about other memories tomorrow, like bioelectric memories. But in my research, I used to always focus on epigenetics and development. So I offer this part of the whole picture. But I believe that all memories can be distributed across several levels, and they transmit signals from, like, during time, from one to another, right? So I will end with the quotation from Ewald Hering, and thank you for your attention, and I will be curious whether you have some criticism or suggestions or questions. Thank you.
Thank you, Jana. So we have about 5 or 6 minutes for questions, and then we'll have a break after that. So if you wouldn't mind not closing your screen share, we can see everybody.
Yeah.
There we go. Thank you. And any questions, comments? Jana even invited criticisms. Marguerite and then Catherine. Marguerite, please make sure you unmute. I might be able to do it for you. Ask to unmute. Did that pop up anything?
Yeah,
There.
okay, I'm on now.
Yeah.
Yeah, thank you very much for that, Jana. I had a ton of questions, but I'm just going to ask you one. Given the plasticity of the genetic adaptations that you were talking about, can you describe a bit more what happens when the induced memories clash? So, for example, you have a genetic memory that's inheritable. As you say, the epigenetics, now we understand, are inherited, that clashes with new environmental information. So that's one clash that could happen. A different clash that could happen is when the environmental information itself is itself clashing. So, for example, within the current climate crisis, you can go from drought to flood just like that. So what happens in those individual cells with memory? Because you talked about forgetting, and I wasn't really clear what you meant by forgetting.
Yeah. Yeah, because what can happen is environmental mismatch. Yeah, when, for example, and these are like nice examples, specific examples, when there is already some epigenetic evidence that, for example, if the mother is fed by a good diet, I will better start with famine. Yeah. So, for example, if there is famine, not enough nutrients in the environment, there comes in our uterus when we are pregnant, there comes, and it is perhaps in different species of mammals as well. There comes this sort of thrifting phenotype. Yeah. So the metabolism of the developing baby is within the first 3 months of its development set up in a way it should store more fat. Yeah, every energy it consumes, it should store into fat. But which happens, for example, when there is a war. Yeah, and there are some good studies on that. But then when the war ends and for example, when the famine also ended, there was an increase of obesity in these populations, which were kind of starving for some period of time. So that's an example of how some memory can be non-adaptive in the next generation. Yeah, because the developing embryo was set up in a certain way. And the clash as such, that will be this clash between what is remembered and the future environment. But you asked also about forgetting, right? So there is this big question of what is forgotten. We used to believe that almost every epigenetic modification is erased during development, at the beginning of the development, at least in mammals and not in other organisms. But we believe that there is this Weissmanian barrier, right? And that everything is erased. But now it's shown that a lot of environmental cues and their marks are remaining in the germ cells, like in sperm and eggs. And for example, in the presence of various RNA molecules, which can further propagate the modification on the DNA and so on. But I mean, from the biological point of view, it's really hard to tell how the organisms decides what to forget and what not to forget. But I think if the environmental signal is strong enough, and it takes some time, for example, in stressful environments, it's shown that some of these signals are to be remembered up to the third generation, for example. Yeah. But if you have a shorter exposure to some event, it won't be remembered. So there is evidence for some selected influences which are being remembered. Not everything will be remembered.
Thank you, Marguerite. Catherine, we have about a minute. If you can ask a quick question.
My question can wait, but I just want to throw out there, thank you, this is beautiful. And my question is mostly about the relationship between the internality and the externality of the environment, and where exactly that boundary is drawn. Scott Gilbert talks about the environment as both a selector and an instructor. And that's where we get to the idea of umwelt, which is sort of an interface between internal and external worlds. Just something to think about. If you have a quick answer, that would be great.
Yeah, I actually published a paper with my student where we actually question the boundaries of an organism, and therefore what is internal to it and what is external to it. So I may send it to you, but it's sort of a processual approach towards the closure, organismal closure, which sometimes cannot be delineated strongly, and yeah, so that will be my answer.
Perfect. Thank you. I'd love to see it.
Thank you.
Great. Thank you, everyone. So we're gonna let's take a 10-minute break now, and we'll come back at the top of the hour, 11 a.m. Pacific. That's in 10 minutes, and get started with our next presentation by Marcus Wild. So see you in 10 minutes.